Good evening, ladies and gentlemen. It's my pleasure to welcome you here. My name is Mike Radford, and for now about another hour, I'm chair of the two organisations. It's a great pleasure to be able to have this meeting to celebrate the activities, the contribution of both charities, UFAW, the Universities Federation for Animal Welfare, and the Humane Slaughter Association. You may feel that they are slightly strange bedfellows, but the common thread between them is that they are both doing a really important and unique job. So far as the Humane Slaughter Association is concerned, I'm sure you will agree with me that it is absolutely essential that while animals are killed for food, that it is done in the most humane way possible. Good life, good death. And the Humane Slaughter Association has a unique expertise which it brings to the industry based on scientific evidence. And similarly, UFAW, as you will be aware, gives grants, underpins research, but increasingly is seeing its role as to disseminate research, to explain its implications to the public, to the media, and to policymakers. I said that I was standing down as chair. May I introduce you to Emma Louise Singh, who is she's been a trustee with us for a little time now, and she is taking over as chair, and I'm happy that the organisations will be safe in her hands. And now I'd like to hand you over to Huw, who will do a bit of housekeeping and then introduce you to the work of the two organisations.
Thanks, Mike. So, yeah, just a tiny bit of housekeeping. We do have a photographer taking pictures tonight, as you probably just noticed. If you don't want us to use your photo, just let us know and we can take care of that. Please wear your badge at all times, and as we said earlier, the emergency exit is out and round to the right through the entrance that you came in. And later on tonight, if you want to talk to any of our staff about the work that I'm going to tell you about, we've all got red name badges, so come and talk to us if you want to know more about our work. As Mike said, it might just be worth explaining a little bit about the relationship between these two charities if you only know one of us and you're sat in here wondering why we hold joint AGMs. So what we like to think of ourselves as sister charities. We share trustees and senior management. I'm chief executive of both, for instance, but they are financially independent, separate entities. So I hope that makes sense and explains why what I'm going to do tonight is give you an annual update from UFAW and then swap hats and give you an annual update from the HSA before hopefully getting to the good bit where you get to have some drinks and canapes and talk to colleagues. I'll try and make sure we get there about quarter to eight o'clock at the latest. So I'll put my UFAW hat on first and tell you a little bit about what UFAW does for those of you who don't know us very well, and also what we've done over the last year or so. So UFAW is a charity that's really focused on animal welfare as a science in the service of animal welfare, the tagline there. And our vision is a world where the welfare of every animal affected by humans is maximised through a scientific understanding of their needs and how we meet them. And we try to bring that about by helping discover what matters to animals, developing scientific solutions to animal welfare problems, and really importantly, getting the evidence out there so that people can actually do something with it to actually improve animal welfare. And in practice, that means supporting scientific research through grants and so on, but also supporting the researchers who do animal welfare science. Animal welfare really is becoming established as a scientific discipline, but it's still not easy to become an animal welfare scientist and establish a career as one, and we're really keen to support the people who do that work. In terms of dissemination, we run scientific conferences, we publish a scientific journal, a book series. We have lots of information resources on our website aimed at experts and the public. And cautiously, we do some advocacy. When we really know that the science argues for a particular course of action, we will go out there and make the case for that. So I'll tell you a bit about all of those activities over the next 10 minutes or so. We fund research grants, ranging from really quite small ones, a few £100 at the lowest end, up to, in some years, six-figure sums for multi-year projects. Last year, the year we're talking about, 2023-24, we funded 13 grants, and I've picked out a couple of highlights just to show you the diversity of what we support, which is really anything that's excellent animal welfare science that we think might have an animal welfare impact. So a couple of examples is a recent project in Denmark that we're really interested in, looking at whether recently introduced methane-reducing food supplements for dairy cows impact their welfare. And this is one of those really interesting questions where environmental sustainability might be in conflict with animal welfare, because there's some suggestion that these supplements, which reduce methane and therefore are good for the environment, might impact these cows' welfare. And that's going to be a hard decision to work out which to prioritize. But the first thing is to do the science and find out what the welfare impact is. So that's something we were easily convinced to support. Just to give you something that's really diverse, we've recently funded something looking at trying to decode cats' purrs and discern whether there are differences between purrs that occur due to animals being content and animals having poor welfare. So moving on to supporting animal welfare scientists, and a real focus of that is supporting the next generation of animal welfare scientists. And one of the ways we've done that for a long time is to fund undergraduate, typically, sometimes master's students, to do a short research project as their first introduction to doing real animal welfare science. So we fund these projects, which have been instrumental in establishing some people's careers in animal welfare science. I think there's at least one person in this room whose career started off with a UFAW student scholarship, and another one who started off doing an HSA scholarship. So they're really important to getting people enthused and starting to work in animal welfare science. And what I wanted to highlight about last year is not just the diversity of species and scenarios that these students have been doing projects on, but the international nature of these projects. You can see a really great representation of countries in the global south and elsewhere that may not have a long tradition of doing animal welfare science, but we are super keen to support animal welfare science worldwide, because some of the biggest problems are not on our doorstep, but we can really help tackle those problems by developing animal welfare capacity around the world. One of the problems with that ambition has always been that we would get amazing applications from super keen students around the world, often in the global south, who just didn't quite have the support to put a really well worked up proposal to us or to do a project. Sometimes there's just not the capacity to support those students locally. So over the past few years, we've been running a mentoring scheme where we pair those really keen students up with a slightly an early career researcher, but someone slightly further on in their career elsewhere in the world, who can support these students to put together a good application and do a good project. And it's been a really amazing scheme that's obviously been hugely beneficial to the students who are supported, but actually is really beneficial to the people who mentor them as well. It's often their first experience of supporting someone to do research, and they get a huge amount from it too. And that's a quick quote from one of the students. Further emphasising the international nature of our work, we have what we call a links scheme, which is a network of universities around the world where they're actively involved in animal welfare, and where we have someone who represents UFAW and our work at that university. And that's been growing quite rapidly over recent years. We now have 157 universities in 50 countries, and the real emphasis, again, is on growing that network in the global south. So moving on to advocacy, which is something you may be less familiar with us doing. we're not like a lot of other animal welfare NGOs who have an opinion on a lot of subjects, but we do have a strong opinion on issues where we understand the science, and particularly where we've been actively involved in developing the science. And I just wanted to pick out one example, which is the move to ban glue traps for so-called pest rats and mice. So in 2022, we funded some research which looked to assess the severity of welfare impact of different ways of controlling rats, which I was also involved in as a co-author, and so was our former chair, Alex Simmons. And that research pretty clearly identifies that just about the worst way to deal with pest rodents is glue traps. And that research was cited in some of the devolved authorities' decisions to ban glue traps. We now have effective bans in England, Scotland and Wales. So use, at least by members of the public, is now prohibited, and it must be licensed by pest controllers. So that's a real win, and something we were prepared, alongside lots of other NGOs, to advocate for, because the science is unequivocal, and we're strongly involved with generating that science. We're still working on that. Literally yesterday I signed a letter to Baroness Heyman, along with other NGOs, urging a ban on the sale of glue traps, because we now have this paradoxical situation that you, I think, can get up to 5 years in prison for using a glue trap, but you could probably buy one in your corner shop tomorrow, because we haven't banned the sale of them. So we'd really like DEFRA to consider moving towards a ban on the sale as well, and hope to see progress on that. But importantly, I think the very UFAW part of this story is that we would also say that that story is far from complete, because lots of the other methods are pretty inhumane as well. And what we really need to do is understand a bit more about the alternatives, and ideally develop some better ones. And we're particularly concerned that a lot of people advise catching rats and mice and releasing them, which we suspect is, although it sounds like a very humane approach, probably doesn't end well for the rats and mice that are caught. And we'd like to get some evidence about that, for instance, and that's something we could probably support if we had a bit more funding. So I'm going to move on to dissemination. One of the fundamental ways we do that is publishing the scientific journal Animal Welfare. We've published it for over 30 years, but in this year that we're talking about, we made a really fundamental change, which is that we took the journal open access. So prior to that, you had to pay to subscribe to read all this really important science that we were publishing, and that meant lots of people couldn't access it, including some policymakers. So we partnered with Cambridge University Press. The journal is now fully open access. Anyone can read it online anywhere in the world, which we're absolutely delighted with. The one downside of doing that is that you switch to a model where the author pays to publish, and that can be a barrier for some people to publish the science. But we've got a really excellent deal with Cambridge University Press, who are a not-for-profit publisher, which means that we can offer waivers. We offer an automatic waiver to authors from the Global South, and for anywhere else in the world, if authors don't have recourse to funds from a grant or whatever, they will get a waiver. So the only barrier to publishing in animal welfare is whether the science is good enough. So we're really pleased with that. And Animal Welfare publishes a growing number of papers, 48 in the year we're talking about. It will hopefully be more this year, on a whole gamut of subjects, from fundamental reconsiderations of the conceptions we have of animal welfare to very species-specific, scenario-specific, but very impactful studies. We also publish a book series, and in 23-24, we published 3 new books. An excellent book on wildlife ethics from a group of philosophers and ethicists, which tackles some really prescient issues around wildlife management and conservation, particularly relevant as we talk more about our impacts on wild animals and rewilding and so on. A new edition of David Fraser's wonderful book, Animal Welfare, The Science in Its Cultural Context, which the first edition was the book that I read when I decided that I was going to take animal welfare seriously as a scientist, and we're delighted to have dramatically updated version of that. And finally, speaking of new editions, the UFAW Handbook on the Care and Management of Laboratory Animals and Other Research Animals, which trips off the tongue, has been published since 1947, and this year it fell to me and my colleague Claire Richardson to edit the 9th edition of that book, which is an enormous book with 51 chapters covering the welfare of more or less every species that's used in scientific research. And just at the bottom there, we've got one that's coming up this year as well. So still publishing lots of books. Another central way that we get the science out there is scientific conferences. We ran an online conference in 2023. As you can see, that had a really excellent global reach as well, 63 countries, 467 participants on the day. It's all online and you can watch it back, so if you missed it, don't worry. That's a really excellent way for us to get animal welfare out there and to share knowledge between scientists. We have very open meetings that are on any subject. It's really just the best animal welfare science, and it brings together a really diverse range of people to share knowledge. In 2024, we ran our conference in person. We run them in person every other year, but also stream them online even if they are in person. And this year we were in Portugal, so we had 170 odd people in person in Portugal, and many online. So that was another excellent opportunity to share knowledge. And finally, I just wanted to point out that we also do much more focused meetings, often with some of our excellent partner organisations that we work with. And I just thought I'd highlight this one because it shows that we don't shy away from difficult subjects. This is a semi-regular series of meetings we hold with our colleagues at the Swiss Federal Food Safety and Veterinary Office of the Swiss government on humanely ending the life of animals, where we looked at the scientific understanding of how to humanely end the lives of animals when humans have to do that. Not just slaughter of food animals, but euthanasia of companion animals, killing research animals at the end of their use in science, control of so-called pest species. And this is a really excellent opportunity to share the latest science, but also to work with the FSVO to look at how that might turn into policy or guidance, which makes real-world change. We also run some excellent collaborative meetings with the RSPCA and the Wild Animal Welfare Committee and lots of other collaborators. Just to let you know, these are the dates for our conference next year, which will be online, so please put those in your diary if you're interested, and we'll be calling for abstracts too for those. So that's my really quick summary of what UFAW's been up to this year. There's just one other thing I wanted to talk about, which is that we indulged in a bit of introspection as well. We did a stakeholder survey of both our members and other stakeholders, which I hope and suspect lots of you completed earlier this year, so thank you if you did talk to our market researchers. And we asked what you thought of UFAW, essentially. And the good news is that people really value what we do. This graph on the right there shows us what's called the Net Promoter Score, which is basically asking people how likely they are to recommend UFAW to their colleagues. And that's a really high score with very little negatives in it. And the market researchers tell us that's a pretty impressive score for an equivalent organisation to us. But you did tell us things that we need to do better, that we could do more to support and represent the animal welfare science community, the scientists and the people who use animal welfare science. And we'll certainly be looking to do that. And we need to be clearer that we're international. I've told you lots about our international work, but some people still have a sort of vestigial memory of us as being very UK-centric. And I recognise the irony of saying that while I'm talking to you at the Royal Society in London. But we do need to make clear that we're international and that we're not solely focused on universities, that you can apply for funding for us no matter where you work, for instance. So we'll be looking quite hard at how we communicate that and how we design our membership model in future. We certainly won't change our focus on animal welfare science, but the way we go about promoting it will change, and the way we talk about ourselves will change over the next year and a half, as amazingly we approach our centenary in 2026. So that's my really quick, I hope, introduction to what UFAW does. What I wanted to do next is just let you hear two researchers who support who we've supported to do some really fascinating research. And the first one is a fantastic project from Daniel Mills at the University of Lincoln, which we were really keen to support because it asks a question that we've asked a lot, which is, why is it that you can tell people lots of information which tells you exactly what you should do in terms of animal welfare, and yet people will, in full knowledge of that information, will go ahead and make the wrong choice as we would see it. So when this was proposed to us, we were really keen to fund it for selfish reasons, because we'd like to take this information and use it to do a better job ourselves. And we also thought, it would be incredibly useful for the wider animal welfare community. So over to Daniel, who can tell you what he found out.
And just before I start, I'd like to thank UFAW for sponsoring this. And I think one of the things I'm passionate about with regards to UFAW is they focus on the science, and it doesn't matter how uncomfortable it is. They're not afraid of the truth, they just want to get there. So I was really pleased when they agreed to fund this. It's something I've been chatting about with a number of colleagues for quite a while, to try and sort of work out what's going on, why aren't these campaigns working. Just before I start, and before Beata starts the clock on me, I'm a vet by first degree. I work in dealing with companion animal behaviour problems. This piece of work was undertaken by quite a large team from across the UK, people working in marketing, as well as psychologists, etc. So whilst I'm here speaking, this is very much a group effort. So I'm sure you're all aware that brachycephalic dogs have a range of health problems, and there have been campaigns aimed at trying to manage those problems. And those campaigns take various forms, from just raising awareness to pointing out sort of the problems of trying to ban their use in advertising, and the issues that can arise. Despite that, when we put the grant in, we were seeing over 1,000% rise in registrations of French bulldogs over a 10-year period, and over about 250% rise in registrations of bulldogs. So we've got the campaigns, and it seems to be getting out of control. Now, one of the things that I'm interested in was a phenomenon known as pedomorphosis. When you show people these two images, you ask people which one of these do you actually prefer. And a lot of people will go for the image on the left rather than the one on the right. And there are a few subtle changes. Not only is it slightly broader, it's got slightly wider eyes, it's got slightly higher forehead. And these are all features that we often associate with children, and they seem to have this subconscious effect on us that encourage caring in humans. But one of the things that I've got increasingly interested in is also signs of disability seem to have that effect on a lot of humans as well, and this tottering gait. So, we think of the toddler tottering along, and because they're about to fall over, we find it sort of quite attractive, but unfortunately that finds its way, and this was taken from a welfare charity I visited in America recently, and they were proud of the tens of thousands of pounds they'd spent on surgery of this bulldog. I must admit I was pretty horrified. It's completely crippled up with this, and this emotionality behind everything. So that's what pedomorphosis is. Anthropomorphism, I'm assuming everybody knows what that is, the danger of sort of putting human thoughts into animals' heads. But we have to recognize that companion dogs are part of the family. And these pedomorphic features, they will encourage attraction, they will encourage caregiving amongst a section of people, and also emotional closeness. There's a fair degree of evidence in support of that. The question is, does this create biases that affects people's processing of information, and how is that relevant to campaigns? So, as I said, there's been a number of campaigns to try and dissuade people, and the idea behind them, as far as we can tell, is that people need to know about the suffering of these animals. And if they understand that with education, then you'll increase their empathy and altruism towards these dogs, and therefore people won't want to own them because they'll be worried about that. The problem with this is, as we know, humans are capable of being rational, but generally we're not when it comes to animals. So we have our own personal desires and beliefs, and that gives rise to an emotional reaction. When people see things like this, they get distressed. So it's not so much increasing empathy. Unfortunately, when people get distressed, they think about themselves, and that increases what's known as egoistic motivations. So they personally project into the situation and think about the suffering. What would I want in that situation if I was struggling to breathe? Well, then they tend to think about the sort of desire to help. I'd like somebody to help me. So rather than actually discouraging ownership, what we predicted might be happening is that when people see this sort of material, actually it increases desire to help afflicted animals, but rather than reduce ownership, that's converted into these animals need somebody to care for, and I love them, so I ought to get one. And so it has the complete opposite effect to what we might predict. And there's a number of factors that we can sort of use as proxies in this process. So we can look at sensitivity to some of those those pedomorphic features which they might be attracted to. We can look at in the emotional strength or the emotional content of the bond that these owners have with these dogs. And we can look at sort of the egoistic motivations where they put themselves in that the features of ownership. Are they focusing on themselves or the animals? And you can also predict that, well, if they get a dog then like this, they'll become aware of some of the issues, and that's going to create dissonance for them, and they're going to have mechanisms to deal with that. So you get the campaign material, you've got one of these dogs, but at the same time you're getting messaging that you shouldn't own one of these dogs, so people will engage in a range of dissonance reduction strategies. This can be sort of classically forgetting or discounting of information. Well, you can forget about forgetting, because if it's being rammed down your throat in adverts, you're not going to forget it. So we wouldn't be looking for that, but the discounting of information may be important. And you can explore that by looking at things like underestimation of the costs of care or trivializing of the problem. And another strategy that people use is often reframing of information. They try to represent it. So they create other features of the dogs which are desirable and place more emphasis on that, or they blame others. So this is what we aim to do. And it was a mixed method study, and I'm going to rush through because I have only got 10 minutes, but there is a chunk of papers that are going to be coming out as a result of this. And the qualitative work is really important in this, because otherwise we end up just exploring what we already are thinking are the solutions. And there's a lot of information that came out from the interviews that fed into it. But I'm going to focus quickly on the results. So what did we find? Well, what we found were there were some really strong effects amongst the brachycephalic owners. They were more strongly attracted towards brachycephalic features, not just in short-nosed breeds, but in the other ones as well, far more so than the other groups of dog owners that we surveyed. When we asked them about the emotionality and the emotional strength of the relationship, they're much more likely to put a ring round circle G there and see themselves as that much closer than the other groups as well. Much more likely to agree with statements like, my dog is like a child. So, and then if we look at those sort of egoistic motivations associated with ownership, then sort of we've got overall stronger agreement with statements such as, my dog makes me feel important and needed. Now this is quite interesting because, as we'll see later, as I said, some of this work was done with the support of people who work in marketing. And there is this brand and image of being an owner that is very strong within this group as well. There are also some, and that went across both of the, compared to the other control groups. We had two control groups, one which was sort of all other, what we call all other dogs, which is the larger dogs, not the size match controls, and there were differences between that as well. They wanted to dress the dog up more, factors like that, and I want a dog who is dependent on me. And whilst there was no difference to the size match controls, the size match controls didn't differ to the all other dogs. So these people do want to dress their dogs up, and yeah, they want to do things to these dogs that they enjoy as well. So as I said, we had three sort of populations. We had the brachycephalic owners, we had similar sized dogs to the brachycephalics, and then we had the group that we called all other dog owners. And when we looked at the content, then we can see that the brachycephalics, there's a very clear identity and there's big influence of social media as well. We predicted that there would also be signs of dissonance as well, and again, that's clearly there. There's stronger agreement with statements related to the recognition that the dog isn't healthy. So there are signs of active dissonance and disagreements with things like the benefits outweighing the disadvantages. So the experience of owning does have an effect. As far as strategies that they were using, we saw evidence of trivialization, so belief that these dogs are suffering, not enjoying their life, creating other benefits. One of the things that came through on the interviews was that the snoring noise of the dogs was comical. It's a cute feature. Let's reframe that as opposed to my dog is suffocating. And blame shifting. They're much more likely to sort of say that, well, it's something the government ought to be doing this, or the breeders, or even the veterinary profession. It's not the buyer's fault. So what we have here is, in marketing terms, what they call a brachy tribe, or they call it a tribe, and we call it a brachy tribe, which is a group of individuals who have common shared values. And that has important implications, because it's not about information, because this group, as a tribe, they have shared values, they have a love of the breed, they have a sense of belonging, but there's a personal identity issue here that we've got to address. There's a saviour mentality, that I'm seeing myself as saving these dogs. And there is the brand, which is promoted by social media. And they will have dissonance reduction strategies, and that can actually isolate them from professionals and professional support. So just to finish off, and I know that's a quick rush through, but hopefully it's given you a flavour of what is going on here. And when I've explained this to people since then, they've said, yeah, well, that makes sense. Yeah, so we've got to get our campaigning right. So I think a major problem for current campaigns is that they assume the problem is one of knowledge and rationality, and it's not. I'm pretty certain on that. Brachycephalic owners, they are a heterogeneous population, like any population, but there is a significant core, and we're still trying to model some of the data to see whether or not it's first-time owners in particular or anything like that. But there is this significant subpopulation that seem to form what marketing people call a tribe, which has strong links with their personal identity and with the brand. And so any marketing or campaigning has got to focus on changing the nature of the brand and the value placed in that brand if we are to sort things out. And it may be underpinned by these strong subconscious tendencies that are associated with the look of the animals. So we have to change the tribe, the brand, and potentially personal identity features with these dogs. So thank you very much. A bit of a rush through, but hopefully enough to whet your appetite. And thank you once again to you, Phil.
So thank you, Daniel. Because we're trying to rush you through to your drinks, we're going to postpone questions so you can ask them face-to-face to our speakers when you've got a drink in your hand, hopefully, if that's okay. So next, I wanted to move on to one other UFAW-funded project. Again, a subject close to our hearts, which is the welfare of animals used in scientific research. UFAW was essentially founded, amongst other reasons, to have concern for the welfare of animals used in research. And this is a really interesting project looking at an evidence-based approach to refining lab rat housing and husbandry by Vikki Neville, who is also our Early Career Researcher of the Year 2024. So welcome, Vikki.
So thank you, Huw. I've only got 10 minutes, so I'm going to make a really, really quick start. So I want to start with a statement that I think isn't very controversial at all, and that is that laboratory rat welfare is very important. It's really, really important, and it's important actually for a number of different reasons. So reason number one is that because the weight of evidence suggests that rats are sentient, and because we, as humans, have decided to keep these rats in captivity and to breed them, we have a moral obligation to try and minimize their suffering. And actually, to go further than that, we have a moral obligation to give these rats the best life possible. Reason number 2 is that because we think our fellow humans are also sentient, we have a moral obligation to try and minimise their suffering and to give them a good life too. And we know from previous research that putting things into place to try and enhance the welfare of animals in a lab has a positive effect on the staff working with those animals too. So it reduces the risk of burnout and of compassion fatigue. And reason number 3, and maybe even the most important reason for some people, is that good welfare is necessary for good science. So we know that, poor welfare can compromise a whole host of outcome measures, spanning behaviour to immunology to physiology. And so it's really crucial for biomedical science that we work to try and improve the welfare of animals in the lab, especially in light of the ongoing reproducibility crisis in biomedical research. So in 2015, it was estimated that $28 billion was spent per year on preclinical research that was not reproducible. Now, in the UK at least, a good proportion, a good chunk of biomedical research will be funded through research councils, so through taxpayers' money. So we are just wasting taxpayers' money if we do not think about the welfare of animals used in science. Good welfare is absolutely paramount for good science that is better value for money. And now a slightly more controversial statement, although hopefully not too controversial given the audience, and that is that standard laboratory rat housing is unacceptable. It is completely unacceptable. The average height of an adult male rat is about 28 to 30 centimetres. So rats don't spend all of their time standing up, but if you spend any time watching a pet rat or a rat in the wild when they're not sleeping, you'll realise it's actually quite a common behaviour. The minimum cage height, as specified in the UK Home Office Code of Practice for the housing of animals used for scientific purposes, is 20 centimetres. Now, a bit of quick maths, 30 minus 20, tells you that there's a little bit of a problem there. 10 centimetres of a problem. It is not just about the cage height, though. If you just spend a single second thinking about wild rat ecology, you'll realise how abnormal and restrictive these cages must be for rats. They can't run around. They often don't have an area of complete darkness where they can rest. They often can't even hold food with their beautiful little hands. It's really quite bad. But that brings us to the question of, well, what is actually acceptable? Now, I can give you an educated guess about what is going to be best for rats, but ultimately, as you can see, I am not a rat. And rats inhabit an entirely different sensory world from me. Their world is really very different. So because we can only think about refinements from a really human perspective, it's really important that we don't just assume that what we think should be good for rats is actually any good. We need to have good evidence that refinement is likely to be successful to avoid wasting time or money or energy or resources on refinements that might not be successful, or even worse, could be detrimental to the welfare of rats. But what do I mean by good evidence? Actually, that's quite a nebulous term. So for me to feel happy that a refinement was going to be successful, I would want to know that the potential refinement would improve welfare according to multiple rigorously designed studies, that it did so consistently across those studies, but they also did that across context. Now, if you start to pull this apart a bit, you'll actually realise it's quite a tricky question to answer. We first of all just need an overview of what has already been done. So what refinements have been studied, what has and hasn't been done. Then we also need to know about how those refinements have been tested, because there are lots of different ways to measure welfare, potential ways to measure welfare, and there are pros and cons of different approaches. We want to know about the quality of the research. Is the research actually any good? Is good science being done? And we want to know about all the factors that might moderate the extent to which the refinement does improve welfare. And that is where a mapping review comes in. So a mapping review is a transparent and a systematic approach to outline and catalogue evidence. So it's a really useful approach to tackle the broad sorts of questions that we're trying to answer here. So that's what I wanted to do. I wanted to do a mapping review of refinement to laboratory rat housing and husbandry. So I started on this task, I did my database search, which is where I very quickly hit a roadblock, and that roadblock was the sheer number of articles I found when I did that initial search. But UFAW, very thankfully and kindly, came in and saved the day here. I applied to them for some funding to hire some research technicians. I was awarded that funding, and it meant that the whole project was feasible. So the project went ahead, we did screening, another round of screening, until we were left with over 1,000 articles that were included in our mapping review. And so in the interest of time, I'm not going to go through the nitty-gritty details of our methods and all of the results. I'm just going to skip to the good bit, take-home messages. And take-home message number 1 is that so much more research is needed. So the majority of the research focused on really similar refinements, things like just putting a few objects in the rat's cage. Most of the research was conducted on male rats only, and many of the articles didn't implement aspects of experimental design that we know are really, really important for good quality, robust research. So lots more work that UFAW could potentially fund there. Thank you, UFAW. But from the evidence that is available, we know that rats really need good shelters. That is a solid area that provides them with complete darkness, which actually most of the time they're not provided with in the UK at least. The cardboard tunnels you see, they're just not good enough. They also need objects to chew, which thankfully many rats are provided with. But one of the clearest messages that came out of this review was that a one-size-fits-all approach is really, really not appropriate. The review identified so many factors that moderated the extent to which the refinement did have a positive effect on welfare. So the age of the animals, the sex of the animals, the temperature of the colony room, the number of cage mates they had, whether the measures were taken in the daytime or in the nighttime. So complex environments are really, really necessary for these animals. They need options so that they can pick and choose what is best for them at any given time of day. So I think this message about individual differences and thinking about individual differences is a really, really important one. So just to illustrate this, I want you all for the next few minutes to pretend that I am magical. I have magical powers, and I can send you all for 24 hours at least to one of these 4 destinations, and I want to know where you're going to pick. So hands up, who wants to go to A? Who wants to go to B? Who would like to go to C? Okay, that's what I thought. Who would like to go to D? A few people. So you can see that there are some options that are just less preferred, that aren't very good. No one picks C, it's not a great option. But beyond that, we're all going to have different preferences. So some of you picked A, some of you B, there are a few people that also picked D. And, you know, so we all have different preferences, different strengths of preferences. So really that's what we need to think about when we're thinking about rats. We need to think about how we can cater for the needs of all individuals, not just the average rat on an average day in an average setting. And when you think about how that might change as well, so if we go back to this example, you know, I asked you where you'd like to be for 24 hours, but would you pick the same location if you had thought about being there for a whole year? If you had thought about being in these locations when you weren't feeling so well, when you were just recovering from surgery? Again, this is what we need to think about when we're thinking about refinements for rats. So I'm just going to end my talk by saying a bit about how we've tried to put this into practice in the lab, because the question of how we can feasibly implement this with all the constraints of being in a lab is a really, really important one. So first of all, my colleagues and I have designed these cage toppers that you can see in the top photo and the photo on the left. So these are just a topper that goes on top of a standard cage, a standard rat lab cage, and just provides a bit more complexity. So different areas of different sizes, so you can fit more enrichment in there, just gives the rats lots of different options. But the technicians can still conduct checks really easily, they can be cleaned easily, we can still fit a number of rats in the colony room. And we've also started to think about how we can actually track rats on an individual level to understand how individual rats are faring in the lab cage. So to do this, we've put rats into these pet-style cages, a little bit bigger, and then we've made equipment using Raspberry Pis. So we've made a Raspberry Pi camera, you can see the footage of that there, it just sits on top of the cage. We've developed other Raspberry Pi equipment to collect data from these rats to monitor their welfare. And Raspberry Pis are really, really cheap. Lots of people can buy them really cheaply. We can make everything open source so that people can try and do this. And so you can see that we've really tried to build on that mapping review to try and have an impact for rat welfare. So I'm going to wrap up there. The work is already published. If you like a copy and you can't access it, then please just email me. And I'd like to say thank you to lots of people who are involved in various parts of this research, but particularly Natalie, James and Elsa, who were the 3 technicians we hired, who did an absolutely fantastic job. They were amazing. They worked super hard. Of course, thank you very much to UFAW for funding this project. It's a great project, I think, at least. And hopefully a lot of good is going to come out of it. And of course, thank you to BBSRC for funding me. Thank you.
So thanks, Vikki and Daniel. I hope that gives you a flavour of some of the really fantastic science that we support and how it is going to make a real difference for animals. Vikki asked for more money to support her research. It's now time for me to put my charity chief exec hat on and ask you for the money. But more importantly, maybe just become involved with UFAW. First of all, we'd really like you to ask everyone who isn't a member of UFAW to join UFAW. I know we've got lots of members here tonight, and we're very grateful for your membership, but there are also quite a lot of people we've invited who aren't members who we think should be, and we would love to have you. So if anyone wants to join, scan the QR code, or you can talk to Ailey, who's probably out the front, or certainly will be later on. He'll be very happy to sign you up right here and now. But please do consider helping us through being a member. It's the best way to stay engaged and support what we do. And if you really value this work, please do consider donating as well. The name sometimes isn't very helpful, but UFAW is a charity. It doesn't get any government or statutory funding. Absolutely every penny that you've heard spent on that research just now came from individual donors and other sources of support who choose to give us money to do this work, and we really do need that, and we could fund many more projects. We see amazing applications that we can't fund, so we really would appreciate some support. And again, Ailey will be very happy to take a donation out in the foyer, and you can talk to the rest of us about how to donate or become involved as well. All the staff have got red names on the badges if you want to talk to us. So, I'm done with UFAW, so I'm going for another hat change, metaphorically, and I'm going to become Chief Executive of the Humane Slaughter Association for the next half an hour or so. So, the HSA is even older than UFAW. We were founded in 1911. We're well past our centenary. And the HSA specialises in the welfare of food animals or animals raised for their products in markets, during transport, at the point of slaughter, or during emergency killing, which is increasingly an important issue as we see various disease outbreaks. So not just slaughter, but also, as we like to put it, beyond the farm gate. And our vision is a world where all farmed animals are transported and killed humanely. And we try and bring that about by advancing and promoting humane treatment at slaughter, transport, killing of farmed animals, mainly through research and education. So again, just like UFAW, we get people started early by doing student and trainee scholarships. So these are undergraduates or industry trainees doing a short project as their first exposure to animal welfare science. And funnily enough, that's how Daniel got started. So even if it doesn't get people into slaughter research, it sometimes brings them into animal welfare research. As you can see, again, it's a diverse range of projects we funded last year, and geographically diverse as well. And that's again, really important to the HSA. Huge improvements can be made around the world. And again, perhaps even more so, some of these students really benefit from being mentored to get them to do this research, which is quite challenging. I think it's important to acknowledge how challenging a lot of this work can be for people to do as well. At the other end of the spectrum, we fund some very significant research projects over the years to really push forward the development of methods for humane slaughter. You're going to hear later on about the work to test low atmospheric pressure stunning in pigs, but we've also been fundamental to the development of electrical stunning for farmed fish, and a really interesting recent project looking at single pulse ultra high current stunning for cattle, which is a bit of a mouthful, but essentially is a method which could both provide a much better way of stunning cattle, but also be acceptable in terms of halal stunning, so it could increase the uptake of stunned halal amongst the Muslim community, which could make a really fundamental improvement to the welfare of beef cattle. A particular focus has also of late been humane stunning for aquatic species, and in 2020 we spent over £1.9 million on work to develop humane stunning methods for finfish species that are often neglected. So we know how to stun salmon, for instance. All the UK farmed salmon will be stunned, but that's not the case for a lot of the species farmed elsewhere in the world, in Asia, tilapia and so on, for instance. So we funded some work to develop stunning parameters for those species and for crabs, lobsters and cephalopods, octopus and squid. This is all really important work for species that are quite neglected, and those 4 projects are ongoing but coming to fruition. And moving on from that, something you'll be hearing a lot about from us soon is another systematic map or review, taking a similar approach to what Vikki talked about, looking at all the evidence about how feasible it is to stun wild-caught fish. And this is probably the largest animal welfare issue that we can think of, and certainly the largest, essentially untackled one, that we consume over 1 trillion fish per annum, probably closer to 2, caught from the wild, vast majority of which are not humanely stunned. They either die as part of the capture process or they die on the deck of a trawler by asphyxiation. And this review, done by some excellent researchers at Harper Adams, looked at all the evidence that we could take from aquaculture and elsewhere to look at, can we stun fish on boats and solve or start to solve this problem? And the answer is we probably can. There's already work going on, and this review will be published both as an academic paper but also a policy brief to really push this issue forward for the industry and policymakers to start to think about tackling this really enormous, effectively untackled problem. Something you may not know so much about what the HSA does, but it's really fundamental to what we do, is training the industry and providing welfare assessments and advice to the industry. We have really expert technical staff, some of whom are licensed slaughter people, who actually know what it's like to do this work and actually know how to do it in the most humane way possible. And they spend a lot of time training people, looking at abattoirs, at the invitation of abattoirs and food businesses, to provide advice about how to do this as humanely as possible. And you don't hear a lot about that. Most of this work is done in confidence, but it's incredibly impactful. So the welfare assessments we did of abattoirs last year, those facilities processed in excess of 80 million animals a year. So you can imagine that one bit of advice that tells them how to do something a bit better can make an enormous impact on the welfare of those animals. So that's a really important part of our work. We'd be very happy to talk about doing that work for anyone who works with the industry who needs that kind of advice. And it's, as I say, it's really core to what we do. Finally, I'm going to use that evidence-based advocacy phrase again and tell you another issue that we really have been advocating for some change recently. And this is carbon dioxide stunning of pigs. 80% of pigs in the UK or so will be stunned with carbon dioxide, similar in Europe. And we know it's not humane, frankly. I won't mince my words. I'll show you some of the evidence for that. Don't worry, this isn't horrible to look at. This is a pig trying to get to some apples in a chamber that's full of carbon dioxide. You don't have to be an animal behaviour expert to know that the pig's not very happy to stick its head in there, because pigs really like apples, so it really wants to eat those apples. But it doesn't like what's in there. And we know from this and much more quantitative evidence that pigs are strongly averse to carbon dioxide. But that's the way we kill 80% of our pigs. We lower them into a high concentration of carbon dioxide to stun them before they're killed. So we know that's not humane. We know we should be doing something else. that video, if you notice, was from 1992. We've known this for a long time, and everyone's been saying we should do something for a long time, but also what people would say is we need to develop something new, and what about X, Y, and Z? But no one actually did very much about it. And one of the things that was often talked about a lot was something that genuinely is, or could have been, a very promising alternative: low atmospheric pressure stunning, which is a method that's now approved in Europe for stunning poultry, where you simply reduce the atmospheric pressure in a chamber and it will render animals unconscious, stun them, by depriving them of oxygen. And lots of people would say, oh, this could be an ideal way to stun pigs. Let's wait and see whether that ever works. And nobody was actually doing anything about it. So in, I think this was 2018 from asking Jess, it seems more recent, but we joined forces with DEFRA and put our money where our mouth is and funded a really major research project to test this hypothesis, to actually expose pigs to this method and see whether it was going to be the humane alternative. And I'm delighted to introduce Dr. Jessica Martin from Newcastle University, who was part of the research group led by Dorothy McKeegan at Glasgow, who actually carried out this research, and she's going to tell you a little bit about that now. Thanks, Jess.
Yeah, so thank you so much for inviting me to talk here today. Like the others, it's 10 minutes, and I've got a 2-year project to cram into 10 minutes. But one thing I just wanted to say, because again, it's about advocating for what we're here. So Humane Slaughter Association has been instrumental to my career. They funded my PhD and they have funded my work ever since then. And they've enabled me to stay in research that's in end-of-life experiences for animals. And not only have they given the funding and the support that's there, but it's also the networking of being part of a group that provides support and knowledge in an area that is incredibly hard to work in, and there's not many of us. So I just wanted to do that plug before you start my time. So Huw's already done a summary of what the issue is, so I'm not going to bother speaking too much about that. But as Huw said, it's a fact. CO2 is painful over certain concentrations, and in CO2 stunning of pigs, pigs are exposed to that while they are conscious. It's not up for discussion anymore. We know it, we've known for a long time. So the number of pigs that were slaughtered in Europe last year was roughly about 220 million. And if you look at the percentages of the countries in Europe that reported whether they were using CO2 stunning or not, these are the percentages that were stunned with CO2. So if we take an average of those, roughly from the ones that have reported it, we can assume about 60% of those animals are CO2 stunned in Europe last year. That equates to 132 million pigs. That are the numbers we're talking about. This isn't a small number. These are huge impacts on a large number of animals. Now, we won't talk about Brexit, but if you add in the UK, as of last year, we had 90, it's actually up to 90% now. So if you add that on, that's 141 million pigs in 2023 for Europe and the UK that were killed with CO2, which we know is painful and causes severe respiratory distress. So we know that. We need to come up with the alternative. So as you said, the potential alternative was hyperbaric hypoxia, or low atmospheric pressure stunning. And if you're bored one afternoon, what you can do is go onto YouTube, and there's some very funny images and videos of people who are put into decompression chambers, and they're not given oxygen initially to help them as that oxygen level works. And the reason why it had this potential is because it's effectively insidious in people. So people don't know that they're becoming hypoxic, that the oxygen's dropping, and they actually get really euphoric and a bit giggly and a little bit silly. And when you tell them you're about to pass out, you need to put the oxygen mask on, they kind of smile and just sit there. So that was what we were working with, what we thought might be the way to take this forward. There's also been a lot of work done for pilots. So when you're on an airplane, when they say fit your mask first, if the pressure drops, don't wait and fit somebody else's first. It's because you're probably going to be cognitively impaired by the time you then try and put yours on, and you'll be unconscious. So there was a real potential of where this could go. And it's like basically going up in altitude, and that it's not an actual decrease of the percentage of oxygen, it's a decrease in the partial pressure. So all it's doing is that partial pressure means the available oxygen you can breathe decreases. So where this started, as Huw has said, is actually it was originally looked at in birds, and this was led by Dorothy McKeegan at the University of Glasgow, and then myself when I was still at University of Edinburgh and Rosalind Institute. And basically, these are the papers that were published alongside that, and it took several years where we did a substantial and very rigorous animal welfare assessment to basically unpick whether or not this was a humane alternative. And that led to that method, after being going through a very vigorous process with EFSA of it being reviewed. It got added to EU law in May 2018. And collectively, what this shows and what they concluded was that low atmospheric pressure stunning is equivalent to stunning with inert gases, so nitrogen and argon, i.e. it is better than with carbon dioxide. So even though carbon dioxide stunning with birds is not considered to be as big an issue with the pigs, there are higher welfare alternatives that we need to be focusing on. So this led to the project that DEFRA and the Humane Association funded, and that was led by Dorothy McKeegan at Glasgow, myself at Edinburgh, and Emma Baxter at SRUC. And we had three main objectives, and I'm just going to go through them very, very quickly with everybody here. So the first one was trying to identify the decompression profiles that we were going to look at. So we had what we did in birds, but birds aren't pigs, shocking. You know, one's a mammal, one's a bird. And then we've also got a lot of the literature that's in from the work that's done in people, so pilot training. So we basically, through a systematic approach, identified a region of interest of how fast we wanted that decompression to go that would be considered not painful in people, but wasn't going to take half an hour, therefore would be completely impractical in a slaughter situation. So there are constraints with this. It's got to be fast enough that it doesn't slow down the line speeds in a slaughter plant situation. It's got to work in the environment it's working towards. So we had this region of interest, and it meant that we came up with a range of decompression profiles that we were aiming for and the estimatings around them. So we took those forward, and effectively, in the first experiment where we did this in anaesthetised animals because it had never been tested in pigs before, so again, protecting their welfare, we did this in unconscious animals. We showed that LAPS was effective at killing pigs, and it was effective in all the decompression curves that we had. And what you'll see here is the latency of how long it took them to stop breathing. So the slower decompression took the longest, so they stayed alive for longer, while the faster ones, it was quicker. And when we looked at the pathology results and what we could see from the physiological outcomes, basically the slowest one was going to take too long, and the fast one, there was pathology concerns that there might be a bit of, the decompression might be having effect on their anatomy. So that meant we took that information forward, but that study was then published in these two papers that you can see here, and that's what out. But it meant we had that range of where we could go and what we could take forward. So objective 2 is the animal welfare bit, because the first bit, they're anaesthetised. You can't really conclude anything about animal welfare. So this was the animal welfare bit, and I'm really proud to say that this was one of the most rigorous assessments for animal welfare on a stunning method. And it's in the bird work, it still is to date the only method that's been added to the EU directive for a new stunning method, because the amount of assessment that we require now is so much higher. So what we did is we combined all of these different measures, so looking at spontaneous behaviour, EEG, so that's electrical activity of the brain. We employed pharmacological interventions, so using anxiolytics, so anti-anxiety, pain relief medication, to see if these behaviours or the physiological effects changed. We also were looking at the pathology, and then the newest one we were looking at were behavioural paradigms, so actually getting the animal to tell us something when it was doing an operant test. So we were comparing LAPS with CO2, and we also had a sham treatment where we just put them in the LAPS chamber, but we didn't turn it on, because we wanted to see what being in a novel environment would do to, and whether or not that also added any aversion. And then we also had the interventions of the pain relief and the anti-anxiety medication. So this graph here just shows you the decompression profiles and the CO2 in terms of the percentage of oxygen that's available. You don't need to worry about the numbers. The thing I want you to take away from this is LAPS takes 10 minutes for one cycle. CO2 is way less. We're talking a quarter of that time. So the time for an animal to perform something in its animal welfare assessment in CO2 is much shorter, and LAPS was a lot longer. So the results of that assessment were, for this work, was that for LAPS there was 100% that the pigs were killed at the end of that 10 minutes. For CO2, it was 55.6% of them were dead at the end of the CO2 stunning. Now, that is because it is a stunning method, and in theory they would go on to be bled, but we had reflexes and recovery when they came out of that very, very quickly. So in terms of a fail-safe, the recovery time when they come out of that environment can be very, very quick and has animal welfare concerns. So the other thing that it showed when we looked at the EEG data was it meant we got the time of how long does it take for the animal to lose consciousness. Now, obviously, in a cycle that takes 10 minutes and is much more gradual, it took longer than CO2. So there was a longer period of time that the animal might experience painful situations, and that's what we expected. It's a more gradual approach. The key things were we saw aversion behaviour in all 3 treatments. We saw it in CO2, LAPS, and in the sham. Pigs just don't like new places, and they get frustrated. If you keep them somewhere new for more than a minute, they're really inquisitive, boisterous animals, and they just start to get annoyed. So whether or not you're doing anything negative to them, putting them in a novel environment and restraining them is in itself annoying to them and has animal welfare impacts. So some of the key behaviours that we saw that we need to be to be aware of was in CO2 we had an increased number of escape attempts. That was the highest number compared to LAPS and within a sham. But in LAPS we saw more head shaking or head low behaviour. We saw more respiratory distress in CO2. We saw more head tilting in CO2. And also with the pharmacological interventions, what we did was, because again, when you look at spontaneous behaviour, it's quite difficult to understand the motivation of why an animal's doing something. So we took a very mechanistic approach of, instead of putting a motivational drive saying escape attempt, we wanted to just describe what the animal was physically doing, and then we used these drugs to see if these changed. And what it meant was we identified additional behaviours that nobody had recorded before that could be to do with pain and anxiety in these types of environments. So head low, for example, was to do with pain, and gasping behaviour, which is around respiratory distress, went down with an anxiolytic, which could link to things like air hunger. So the second thing that we did is to sort of do the full round view of our animal welfare assessment was we wanted to do the operant task, so the behavioural paradigm work. So basically what this involves is where you trained the pig that if it pushed the blue button, it could leave that chamber. So it learned jam works really well. If it pressed the button, it can leave and it can go. Then what we did while it was in that box is we then tagged a red light with a food reward coming, so it knew, red light, I'm going to get a treat. So it always knew, red light's coming on, food's coming. If the red light's not on, I'm not getting a treat. And then what we did is we extended the amount of time that red light was on. So it meant that the animal would be getting a delayed reward. It knew if it waited there and the red light was on, eventually a reward was going to come. So the idea with this is then what you do is the environment that they're in is then you change it. So you either put CO2 in or you decompress it, but the animal is not doing something that you're trying to work out what the motivation is. You're basically allowing that animal through this trained behaviour to go, I don't want to be here, I'm pressing the button, let me out, or it's not that bad, I'll stay here and wait for this reward. That's the idea behind it. So when we took that forward, basically we showed that, shockingly, pigs are really smart, and the array of tricks you can give them to get them to do these things is brilliant. But they learnt the task for this aversion test. So basically, if they did any orientation towards the bowl or the light, that means they're looking for the food reward. They're pressing the button or orientating to the door, which is to leave. It's because they want to get out. So we had an increase in bowl and light orientation in both LAPS and CO2 at the very early stages. So they were willing to wait right at the start. But you got an increase in button pressing to exit in both LAPS and CO2 within 5 seconds of the chamber, either the gas going on or the decompression starting. So, and then after that period, they stopped engaging with the test. And that's because it was so aversive being in there. They just, telling us to press a button, they just wanted to get out and do extreme behaviour escape attempts. The other thing is, obviously, as time goes by, we're dropping the oxygen. So they're becoming cognitively impaired. So the period that they're able to do this task is limited, and you need to be aware of that and how they perform it. The last thing that we did was looking at the pathology and the meat quality. And again, it's giving that all-over picture of what it can be. So actually, in terms of comparing CO2 with LAPS, there was no significant differences between the stunning treatments on meat quality measures. It didn't have a significant difference. But we did see pathology changes around congestion and hemorrhage in both treatments. So in lungs and things like that, you saw a lot of congestion with CO2 and hemorrhage. But the most concerning one was actually to do with the tympanic membrane, so the eardrums, where we saw in LAPS, 60% of animals had their eardrums damaged or completely ruptured. Now, in a decompression environment, we don't know if that's happening while it's decompressing or once it's recompressing. But and it's really hard to work out. We tried putting cameras into the animals' ear canals when they were anaesthetised to see if we could work out bits and pieces. It's very difficult to do. But so conservatively, from an animal welfare point of view, let's assume it's happening while the animal's conscious, worst case scenario. So there were big concerns with that. And that also tallies with the behaviour we saw, like head lows and things like that. So in conclusion, both lapse and CO2 are associated with several poor welfare indicators. And the difference in duration of the cycles actually makes them really difficult to compare. So you've got CO2, which is really aversive, but it lasts, the conscious period can be up to 50 seconds. Or you've got lapse, which might not be as aversive, but it can go on for 6 minutes that they potentially could be conscious for. Who says which is worse or better? That's hard to do. Both drug treatments provided evidence that there's pain and anxiety associated with lapse and CO2, and the aversion testing worked to show that there was aversion in both of these treatments. And again, lapse showed ear pain with barotrauma. And the key thing which often gets asked to me is whether or not slowing the decompression profile down will give an alternative, or changing that profile. If you slowed it down massively, maybe, but it would never, ever be relevant to a slaughter situation. We're talking hours, not minutes. So it's never going to be something that's an option. So in terms of conclusions, collectively, our studies gave the most rigorous assessment for welfare in terms of lapse and showed it's not a viable alternative for CO2, at least not at the rates that would be used in a commercial slaughter or stunning situation. But what I will say with this is, because obviously it feels like it's a negative result and it's a disappointment, these results were the most detailed and rigorous welfare assessment for both lapse and CO2. And it gave more data to show that CO2 is that bad. And it also means that data that's now out there, those papers are published, nobody needs to ever look at lapse again in pigs. The questions are answered. There's no room for error with that. So the money that was funded to do this, yes, it's disappointing it wasn't the alternative, but it gave the answer that was needed to stop everybody waiting to see if it was a potential. And then the other big one was it just further solidified that CO2 is highly aversive and painful, and we cannot wait another 10 or 15 years for an alternative. It needs to happen now. So thank you very much.
So thank you, Jess. That's a fantastic summary of how impactful that research was, even though we didn't get the answer we probably all hoped for. By the way, just to let you know, if you do want to watch one of those YouTube videos of someone trying out low atmospheric pressure stunning, one of them involves Michael Portillo, so I highly recommend that one. So picking up this story, where are we now? We know that CO2 is not humane. We know that we can't use lapse instead. The truth is, actually, we did already know about some other alternatives which had also been considered, but various arguments put in place about why they might not be the ideal. And those include doing electrical stunning better, which is certainly a possibility, improving the way animals are handled whilst they undergo electrical stunning, which is how the 10 to 20% of pigs that aren't CO2 stunned are stunned, or inert gas. And I'm going to go back to one of those videos again from 30 plus years ago. This is a pig sticking its head into a chamber full of 95% argon to get apples. And again, you don't need to be an animal behaviour expert to work out that that's a bit different. And to prove that there really is argon in there, the pig's going to pretty much render itself unconscious in a minute. And even more compellingly, when he gets a bit of oxygen back on board, he's going to go back and have another go. That doesn't prove it's perfect, but that is clearly a whole lot less aversive than CO2. And again, we've known that for a long time, but what we were always told was it'll be too expensive, it'll ruin the meat quality, all sorts of arguments about why you could do a lab experiment, but it wasn't going to translate into industrial, commercial stunning of pigs. And I'm not going to take credit for this research. This was funded by the EU largely, but recently a group in Germany have taken an industrial CO2 stunner and retrofitted it to use argon instead of CO2 for a cost that is estimated to be something like 30 to 50,000 euros, which is not enormous. The gas costs more as well, but the estimate is it'll add €1 in Germany to the cost of a kilo of pork. And importantly, the meat quality is within parameters, so it's acceptable. So that is the point where I do start to be a bit of an advocate, because there clearly are some things that need working out to turn this into a commercial process. It's slower to stun pigs, and we'll have to do some work about how to maintain the throughput. But it's doable in our opinion. So we're very, very prone in this world, and as an organisation that funds science, to keep saying more research needed, but I actually don't think that's the case anymore. What's needed now is some implementation and a bit of engineering. This isn't fundamental science. And for that reason, it's now our position that it's essential that other alternatives to CO2 are adopted without undue delay. And we're really pushing for that change. But we are the HSA, and for that reason, we're working really closely behind the scenes to engage with the industry, to talk to them about how we make this happen, how we might support the engineering work that needs doing, and remove the barriers to making a change. But I'll be really sad if, it won't be me stood here in 30 years, but if someone is stood here in 30 years and we're still trying to get rid of CO2 as a method to stun pigs, then I think that will be a failure on all counts. But I do think we can push this forward now. So that's that story. I'm going to have a pretty radical gear shift now and tell you about one last project which the HSA supported, and this one's going to be presented on video because it's a project we supported in Malawi with the Lilongwe Society for the Prevention of Cruelty to Animals, looking at improving the way chickens are transported by small-scale producers to market in Malawi, where they're typically transported in a quite inhumane way. And we supported a very simple but very impactful project and worked with them to improve things. So I hope this is going to work. I'm going to just try and play a video now for 8 or 9 minutes from Madeline Nyamwanza from the LSPCA. So fingers crossed that you can hear this.
Madeline Nyamwanza, and I'm the Programs Director of LSPCA. I'm excited to join all of you today to give a short report on the progress of our project titled Improving the Welfare of Chickens During Transportation in Lilongwe, Malawi. Introduction. LSPCA stands for Lilongwe Society for the Protection and Care of Animals, and it is an NGO, animal welfare charity organization based in Lilongwe, Malawi. We also work in other cities and districts. Our mission is to raise awareness of animal welfare issues through education, veterinary training, and the provision of veterinary care. We advocate for the implementation and enforcement of policies, legislation, and regulations that promote good animal welfare. We work with the government of Malawi under the Department of Animal Health and Livestock Development and other partners, like in this case, Humane Slaughter Association, to deliver programs that help us to achieve our mission. Moving on to the problem. Malawi consumes an estimated 60 million chickens annually, which is an average of 165,000 chickens a day. And every day, most of these chickens, they're transported for long distances, up to 58 kilometers, while being held upside down, attached to the handlebars of bicycles, and sometimes just a simple plank, a wooden plank, or a log, as seen in these distressing images. This poor handling and exposure of the chickens to extreme weather conditions result in broken wings, legs, heat stroke, and stress. In 2022, the LSPCA collaborated with Lilongwe City Council to conduct a survey which was aimed at better understanding the welfare issues associated with chicken transportation. The survey revealed that at least 10% of the chickens die in transit and at the markets due to these poor handling and transporting methods. It also revealed that 55% of vendors at that time did not provide either feed nor water to their chickens as they awaited to be sold. The survey further revealed that the majority of the vendors was aware or is aware that it is illegal and inhumane to transport chickens in such a way. However, they expressed willingness to adapt to a more animal-friendly system if it is made available. Following the survey, the LSPCA worked together with the government, chicken farmers and chicken vendors to develop more humane transportation methods in the form of chicken baskets or chicken crates. We included the vendors in this process because we wanted to make sure that whatever design we would finally come up with would be easily adopted by the end users, who are the vendors. Initially, we worked with 100 vendors, to which we provided chicken welfare education and enabled access to locally made chicken-friendly crates. And the impact was evident as we saw immediate reduction in the injuries and deaths amongst transported chickens and positive feedback from participating vendors. Although our sample size was small, this was enough for us to know that if we increase the sample size, we would get better impact. And this picture shows the crate that we developed in collaboration with the partners I mentioned earlier. Following the success of our pilot project, we expanded the project with support from Humane Slaughter Association, and the goal for expansion was to educate more chicken vendors in Lilongwe about chicken welfare and chicken transportation, and to train local artisans to make suitable crates for carrying chickens, and ultimately to kickstart a widely adopted new transportation system that improves chicken welfare. So in order to achieve this, we set some project activities. First, chicken welfare training. With the support of Humane Slaughter Association, we aim to train about 350 vendors. So far, we have trained 300 vendors on basic animal welfare, but also focusing more on transportation because this is the main problem we want to address. The second thing that we have done is to source and distribute bamboo seedlings to farmers so that we can promote sustainable bamboo production for future crate manufacturing. And the most significant and biggest project activity is the actual production and distribution of bamboo chicken crates. Our aim is to manufacture and distribute 350 bamboo chicken crates to the trained vendors and to also train 5 craftsmen in crate production to ensure ongoing sustainability beyond the project's duration. And I'm happy to report that so far we have managed to manufacture and distribute 280 bamboo chicken crates to trained vendors, and we have trained 10 craftsmen in crate production. Now, I'm going to show you the progress of our project in pictures. A beautiful picture showing 5 gentlemen just after receiving their new crates. This picture, it shows that the training never stops. So LSPCA project officer was on his way to deliver the crates to all the trained vendors at the market when he came across this gentleman who was carrying chickens inhumanely. He took this opportunity to stop and to explain to the vendor about proper handling methods and also good animal welfare practices. And I'm happy to report that after this encounter, which we see in the picture, this gentleman was later on enrolled into the program and also managed to get his own crates to transport the chickens. This picture shows a typical setup where chicken vendors are receiving training from LSPCA project officer. It is usually very challenging for the chicken vendors to come out to a particular location to receive training. So what we have done is we go into the markets where they are selling the chickens and we just ask for a brief moment with them to conduct the training. This is also another picture showing a crate which is not ideal, and our LSPCA officer here was explaining to the vendors that this crate, while it is a good step, it is a little bit short and the dimensions were not good enough for the number of chickens that the vendors were using. So they were demonstrating here in the picture why this is not a suitable crate. Results of our interventions. There's evident improved chicken welfare during transportation, especially in the areas that we have worked in. There's widespread use of chicken crates, and it is encouraging to note that other vendors who are not part of our project are starting to make their own baskets and crates for use. We've also received support from the government and law enforcers as they have now started to do occasional patrols to arrest offenders. And we are receiving interest from more and more vendors to learn about chicken welfare and to join the program. Finally, I would also want to share a few challenges that we have encountered during the implementation of our project. The main one is that the current design does not protect the chickens from rain and excessive sunlight. So we encourage the vendors to put a thin layer of straw or whatever resources or materials they have on top of the chicken crate so that they can protect the chickens from excessive weather conditions. We would have incorporated that in the production of the crates. However, it was going to significantly increase the production costs. So it's a work in progress. We are constantly working with the vendors to encourage them to use whatever resources they have to try and protect the chickens. The second challenge is that ensuring that all vendors and craftsmen effectively adopt new practices and skills may require ongoing support and follow-up. And lastly, we would also want to expand the project to reach other underserved areas. In this picture, the vendor decided to put an old sack on top of the crate to protect the chickens from excessive weather conditions. And we thought this was quite innovative, and we're also using this as an example to other farmers to show that most of the things that we have in the household can potentially be used to cover the top of the crates in order to protect the chickens. And with this said, I want to thank all of you for listening to me, and I want to thank Humane Slaughter Association for partnering with us to implement this impactful project. Thank you.
So, thank you. I hope that gives you some sort of flavour of how diverse what we fund at the HSA is and how important some of this really fundamental work in the Global South can be. It's a fantastic example of one welfare, in fact. I should say that as Rebecca's over there in the corner, one of the founders of the concept, but animal welfare that can benefit humans as well can be incredibly impactful. So, it's time to do this bit again. I hope you appreciate what we do. If you do, we would really love you to support this work. Again, it's all supported by our members and donors and supporters. And Ailey would be very happy to take your contributions if you'd like to support our work. And also, exactly like UFAW, HSA is a membership organisation. We would love to have you as a member, either if you're involved in this work or you want to support it. And I should emphasise that we also do offer corporate membership, and we're very keen to engage with anyone involved in the industry who would like to be a corporate member. So again, Ailey or any of the rest of our staff would be happy to speak to you. Ailey's over there, she can wave. So, or scan the QR code. We're almost to the drinks time. I just need to say a few thank yous. Thank you to everyone who came. I'm really grateful for everyone coming and engaging with our work. Thank you to the speakers. I apologise for putting you under all that time pressure. Sorry you have to ask the questions whilst having drinks, but you know, we wanted to cram in as much as possible. Thank you to all the staff who've made this meeting possible and done all the work behind the scenes, both the ones who are here and the ones who aren't. The Royal Society for hosting, and one that's not on the list that I'm going to sneak in, but I also just wanted to say a big thanks to Mike as he steps down as chair. He's staying on as a trustee, which I'm incredibly relieved about, but he's done his, is it 3 years? 3 years as a chair, and he's been an incredible support as we've made some real fundamental changes to UFAW and HSA over those years. So a round of applause for Mike, I think.
HSA and UFAW are both evidence-based organisations, and the evidence we've presented to you this evening is the significance of the research that the organisations fund, and just as important, the quality of the researchers. Without supporting researchers, the research comes to an end. And I would suggest to you that if you want to support animal welfare, the best investment you can make is to support research. The only thing that stops us supporting more projects, supporting more researchers, is financial. And it's the research that changes everything. It's the science that changes everything. The more science that is done, the greater our understanding of other species, their capacities, the way they experience their world, and the impact that human use and human treatment has on them. That research changes the ethical debate, and the ethical debate feeds into public policy, and things get done. If you've got money, put it into research. Let us do more. To finance animal welfare research is to be in touch with infinity, because you have no idea where the impact of that research will end. Thank you.
I knew we could rely on the lawyer for a good closing speech. So I won't...
To have the last word.
So thank you again, everyone, for coming. We've got drinks out in the Marble Hall now. We probably have to be out by about 9:30. I think we slightly fibbed when we said 9. So please enjoy a drink on us, and don't be surprised if we come and ask you for some money whilst you're doing it. I can't help it. Thank you so much for coming, everyone.